thanks for having me here at this Science Cafe talk. My name's Ed Lochocki. I'm a postdoc working in Professor Steve Long's group at UIUC, and today I'm going to talk about feeding the future with virtual crops. You might be able to guess from the title that I work in a plant biology research group, and maybe that I do computational modeling of crop growth. So that's what I'm going to be talking about today. I think it's a really exciting area because of the types of problems that we tackle in this part of plant biology and also the methods that we use. And that's going to be the focus of my talk today, is to kind of give you a broad overview and then move a little bit into my own personal research. If you have any questions, like while I'm talking, please feel free to interrupt. I won't mind. Okay. So in my research group, there are kind of two trends that guide most of the work that we do. So one of them has to do with the growing global population. So here's a graph from the UN showing historical data and predictions for the next several decades. In 2020, there are about 8 billion people on the Earth, but by 2100, there are predicted to be about 3 billion more. And with more people comes a lot more demands for resources, and especially ones that we derive from plants. So we get a lot of food directly from plants, but we also get the feedstock that we use for raising animals that we use for food. We also get textiles, and hopefully, at least in the future, we will get a larger and larger share of our energy needs from plants as well. So this is kind of a big challenge for the future, is how are we going to grow all these plants that we're going to need? At the same time, we have to consider climate change. So over here, I'm showing the yearly average global temperature for the last 100 years or so. And you can see that the temperatures have gone up by about 1.5 degrees during that time period. And this climate change is very important when we're considering growing plants, because they're greatly affected by the amount of CO2 in the atmosphere as well as the air temperature. Both these things really strongly affect plant growth. So if we want to make sure that we can meet the demands of the growing global population, we also need to make sure that we can meet those demands in a changing climate. And at the same time, agriculture is a big driver of CO2 emissions worldwide. So we also want to make sure that we're able to grow all these additional plants that we'll need to feed these additional people without continuing to damage the environment. Well, that's probably impossible, but trying to damage it as minimally as possible. So as I mentioned, we will need to harvest more from our crops, and we can try to take a logical approach to figure out what avenues are going to be most promising for increasing our harvests. So one way to look at it is to think about the total crop harvest as the product of the amount of land devoted for that crop multiplied by the harvest per unit area that you get from the crop. And so if you want to increase the crop harvest, there are kind of two options you could take. You can devote more land to crop growth, or you can try to make your crops more efficient. Increasing agricultural area often means destroying natural spaces. If you think about it, there's no free land on the earth that's just sitting around doing nothing. So if you add 1 more acre of farmland, you have to subtract 1 acre from something else. And typically, you're not converting like parking lots or Walmarts into cropland. You're taking natural areas instead. And so this can have a pretty devastating effect on the environment. So we would rather, if possible, increase the yield that we get from our existing cropland. If we go a little bit further, we can try to understand what kind of factors influence the amount of harvest that we get from a particular piece of cropland. And here I'm going to think about the maximum harvest that you could get. under ideal growing conditions. So this means a situation where your plants have all the nutrients they need and they're not being attacked by pests. So both of these other factors that I mentioned and several other ones play a really big role in determining crop yield, but I'm not going to consider them here. Other people are working on them, but that's not something that we focus on in this group. So to understand this equation, it's helpful to think about crop growth as like a process where solar energy is converted into mass that we can use for whatever purpose we want. So the main input here is the total solar energy incident on a particular piece of farmland or ground or whatever during the growing season. So this is kind of the maximum amount of energy that a crop can use. Of course, plants don't usually completely cover the ground, at least not for all of the growing season, because they'll start out very small. So we need to consider the fact that some of the sunlight is not intercepted by the crop. So we multiply the total amount of energy available by the fraction that's actually incident on a crop. And typically, for most crops, this is around 90%, so pretty high, and there's not a lot of room for improvement here. Out of all the energy that's intercepted by the crop, only some of it is actually able to be stored by the crop. So we call this the conversion efficiency, and for many crops this is on the order of 20%. Once we know how much energy is stored by the crop, we can convert it into mass. So we would need to know the energy content of our plant's mass. This number tends to be pretty constant across different crops, and as with the fraction of sunlight intercepted, there's not a lot of room for changing this. And finally, we need to consider how much of a plant is actually useful to us. If you think about a specific plant like corn, you generally care about harvesting the kernels of the corn. And so you want to make sure that your plant isn't investing too much in other parts of its body, like leaves or stems, to the detriment of what you consider to be useful. And most crops, for most crops, this value is about 60%. And so over like the last 100 years, or even longer than that, there have been a lot of increases in the harvested mass per area of various crops. And these increases have mostly occurred by maximizing the fraction of sunlight intercepted, the fraction of useful biomass, maybe not so much this one, but this kind of leaves the conversion efficiency as a big target for a way that we can try to improve our crop yields. So we can start thinking about how to increase this number. So you can ask, one way to start tackling this problem is, how is this intercepted energy that the plant receives from the sun, how is it lost, or how are parts of it lost? There are a bunch of factors at play here. So some of them include the fact that some of the sunlight is at wavelengths that are not useful to the plant, that it can't use. Some of the light will be reflected or transmitted by leaves. And then there are some more like biochemical inefficiencies involved, like during carbohydrate synthesis, some energy is lost. Energy is used just to maintain the cells that are involved in plant leaves. And we can kind of group all of these factors together under the broad heading of photosynthesis, and we can say that our strategy is to improve plant photosynthesis. So to put all this together, here in the Long Lab, we're interested in trying to create plants with more efficient and resilient photosynthesis, with the goal that these plants will be able to store more energy from the sun, and we'll be able to get a greater yield per acre from them. And this will allow us to meet the demands of the growing population using less land area, which will also help to minimize the environmental impact of agriculture. Um, so many of us working in this research group are actually part of a broad or a larger international research collaboration that's called RIPE, which stands for Realizing Increased Photosynthetic Efficiency. And you may have noticed that logo down here at the bottom of my slides. This is where I get my funding from too. RIPE itself gets funding from a couple sources, but mostly, or a big donor is the Bill and Melinda Gates Foundation. So something interesting to keep in mind is that big increases in productivity have happened before. And kind of the main one is commonly referred to as the Green Revolution. But a problem with it is that the technology that came out of the Green Revolution was not shared equitably around the world. And many people, especially small farmers in lower income countries, were denied access to this technology that can vastly improve crop yields. And we are careful to not forget about that, and a big component of RIPE is that we want to ensure global access to any technological developments that come out of this research project, and especially to make sure they're available to small farmers in low-income countries. So personally, I'm very happy and excited to be part of this group because I think the mission here is so important, and I really appreciate that there's kind of like a socially responsible aspect to it. So RIPE has been around for about 10 years now and has produced a lot of research output and some really important breakthroughs. One thing I want to share that I think is really cool that's come out of RIPE is called VPZ tobacco. So this is a research project that was performed by two former lab members, Vanna and Kasia, who used to be part of Steve Long's group but are now professors elsewhere. And it has to do with something called photoprotection. So it turns out that it's actually possible for a plant to receive too much sunlight. This was kind of a surprise to me when I first heard about it, because you would think that more sunlight's always good for a plant. But it turns out that if too much energy is sent into the CO2 fixation pathway, instead of just producing sugar and the other carbohydrates that plants need to survive, they can accidentally create toxic byproducts called reactive oxide species. And so in order to avoid this, plants have developed a protection mechanism. So they can direct some of the incident energy away from the CO2 fixation pathway and into a different route that's called NPQ. So this stands for non-photochemical quenching. You don't really need to know that, but basically what you need to know is that it's a protection mechanism that's activated by high light. And so there's a bit of a problem with this, and you can notice it when a plant goes from, or when a leaf goes from high light to low light. And the problem is that NPQ takes a long time to turn off. So here in this diagram you can see the letters NPQ are the same size, indicating that the NPQ pathway is activated to the same degree. And so this means that most of the light is going into the NPQ pathway instead of the CO2 fixation pathway. And effectively what's going on is that this plant is throwing away some of the incident light that it's receiving, and it's not using it for photosynthesis. And only after a much longer time, somewhere on the order of minutes or tens of minutes, will the NPQ process relax, and now CO2 fixation can increase to its maximum rate for that given light intensity. So you can see here NPQ is smaller and CO2 fixation is bigger. There are many different factors that influence the rate at which NPQ turns on or turns off, but people have identified a couple important ones. So there's a structural protein in the leaf chloroplasts called PSBS that plays a big role. But because it's built into the structure, its levels are always constant, and it doesn't necessarily play a large role in determining when NPQ turns on or when it turns off. So that role is controlled more by the presence of a pigment called zeaxanthin. So you can see here when NPQ is high, zeaxanthin is also high, and then only when zeaxanthin decreases do we get a decrease in NPQ. And if you go a little bit further, you can learn that the levels of zeaxanthin are controlled by two enzymes called VDE and ZEP, which allow zeaxanthin to turn into violazanthin or vice versa. And so this cycle where these two pigments are converted into each other plays a big role in NPQ relaxation. So the idea of this project was that if you can make a plant that has more ZEP than a regular plant, this will help reduce the quantity of zeaxanthin when the light level decreases, so we can get back to this situation faster and waste less energy due to photoprotection. And that's exactly what Vana and Kasia did. They developed plants that actually have higher levels of all three of these proteins, PSBS, VDE, and ZEP. And they verified that the photoprotection mechanism in these plants turns off faster when the plants are switched from high light to low light. And furthermore, they were able to grow these plants outside in a field and compare them to plants that showed regular levels of the VPZ proteins. And they found that there's actually a really large increase in the size, so that plants with these modified genes grow a lot bigger. And so this is super exciting because people have wondered about the role that photoprotection plays in limiting photosynthesis and whether it would be able to increase the yield of the plant for a long time. But this is kind of the first demonstration in a field condition that it can really have a big impact on plant photosynthesis and plant growth. And so you may be wondering, like, why was this done in tobacco? And the answer is that for us, tobacco is kind of a model organism. It's relatively easy to make genetic modifications to tobacco, and it also produces a lot of seeds, so it's very easy to get a lot of plants after you've made a modification. And so the goal is, after you demonstrate that something works in tobacco, you can try to translate it into a more important food crop, such as corn or cowpea or cassava. And of course, in the right project, our ultimate goal is to get real plants that perform better than previously available plants. But at the same time, modeling can play a really big role in this process. And to understand that, it's helpful to look at something that we call in RIPE the transformation cycle. And it consists of 3 steps that we call identify, analyze, and translate. So the first step is identifying traits of interest, and this is the main place where mathematical models come into play. And I'll talk a little bit about that more in a second. The next step would be to analyze them by using genetic engineering to modify those traits in model species. So for the example I just showed, the VPZ tobacco, that's kind of at step 2. And then the third step is to take the most promising modifications that have been identified and analyzed and translate them into the target crops. And in RIPE, these target crops are, I guess I mentioned this before, but maize, cowpea, cassava, those are, and soybean? Maybe I should know that more. But if you think about this process, there's kind of like a dual role for modeling. So as we go from step 1 to 2, modeling can provide the motivation for experimental work. And that did happen with this photoprotection example I showed before. There were some theoretical studies making predictions for how much energy could be gained if you can make NPQ relaxation occur faster. And then the second role is actually we can go back from 3 to 1. And modeling can help us synthesize the results that we get from experiments so we can understand them better. And I want to highlight this kind of role that modeling can play by showing 2 examples of modeling studies that occur at some different scales. These are not actually part of RIPE, but I think they're cool and I wanted to share them. So the first one I want to talk about was performed by a colleague of mine, Megan Matthews. She is currently a professor at UIUC. She was a member of Steve Long's group for a short while, and this was work that was actually done during her PhD when she worked in the Williams group at NC State University. But here in this project, what they did was they looked at a wide variety of poplar trees that all have variations in their genes and therefore variations in the properties of their wood. And so across all these different poplar plants, they measured RNA-seq data to get RNA expression from these plants. They also measured protein expression, and then they combined that with information about how each plant was growing and the properties of the wood that it contained. And with all this information, they were able to apply some machine learning techniques and come up with a model that directly connects the small-scale genetic information with the large-scale properties of wood. And you can see some of these wood properties over here on the right. And the main application of this is for biofuel production, because poplar is a very productive plant. It produces a lot of biomass every year, and the hope is that we can convert that into fuel, I think using like microbial fermentation. A kind of problem along the way is that generally it's possible to convert cellulose into fuel, but not lignin, which is another component of wood. Lignin is very like a hard and durable molecule that helps to give wood its rigidity and structure, but it can be a barrier to using that wood for biofuel. So using this model that Megan and her colleagues were able to develop, you can figure out which genetic changes exactly are expected to produce a certain set of desired properties in the poplar wood. So this can help guide future experiments. So the second one I want to talk about operates at kind of a much larger scale. So this is work done by another colleague of mine, Deepak, who was formerly a member of the Long Lab but is now a professor at the Indian Institute of Technology. And so here for this study, what he did was he used a computational model for sugarcane growth to grow virtual sugarcane all over Brazil. And he's able to do this using weather data that's kind of representative of current conditions in Brazil, as well as those that are expected to occur under several different climate change scenarios. And so what he can see is that in different spots in Brazil, he can predict how climate change will affect sugarcane yield. And this kind of information can be really powerful for deciding at the governmental level like how much resources to devote to sugarcane production for biofuel. Because you might imagine if there's a good chance that your plants won't be productive anymore in 10 years because the weather and temperature is totally different, it might not be good to invest in that. And this is another example of the kind of information that we can get from modeling. So I think these two studies are pretty interesting because they kind of span almost the widest possible range of scales. So at the very smallest scale, we have information about the genes present in a plant. And then at the highest level, we have like countrywide production of sugarcane. And this is part of what I think is really interesting about computational modeling in plant biology, is that it kind of involves a synthesis of many different scales and many different disciplines. Because another way to think about this is that experts in genetics and biochemical methods were required to get all this information about poplar trees. Experts about growing plants and measuring their kind of wood properties were required. And similar people were involved with this sugarcane project because plants needed to be grown in order to parameterize the computational model. And then experts from climate science are also needed to provide the models that we can use for predicting weather patterns in the future. So it's like a truly interdisciplinary field. And if you're working in this field, you have an opportunity to learn about a ton of different things and work with many different people. So that brings me to what I actually do for my research. So I work with a crop modeling software package that's called Biopro. It's free, it's available as an R package, and it's open source. So if you want to use it, please talk to me. I can tell you how to use it. But what I want to do here is point out how Biopro, how the models that we use within it are related to what I was just talking about how they kind of synthesize information across different scales in different fields of study. So I want to kind of go through and tell you how a general Biopro model works. So as you might be able to guess from what I've been talking about, photosynthesis is really important in our research group, and it's also very important for how we choose to model plants. So photosynthesis, one way to think about it is that it's an extremely complex biochemical reaction. It consists of like at least 200 steps. It's been characterized very well. But when we want to include it in a model, we always have a choice of what level of detail we want to include it with. So kind of at a very simple level, we could just use light as an input to our photosynthesis model. At an extremely complicated level, we could include all of the reactions. Here in Biopro, we try to take a sort of middle ground where we use a simplified version of the full biochemical reaction pathway that responds to the most important environmental parameters. So these would be temperature, CO2 concentration, and the incident light, which is symbolized here as Q. And we call this a semi-mechanistic model, which basically means that it does a pretty good job of accurately reflecting the real biochemical reactions that occur along the photosynthesis pathway. One important thing we need to consider is that different leaves within a plant canopy are going to experience totally different conditions, especially regarding the incident light on them. Because if you think about a crop growing out in a field, leaves at the top of the plant canopy are going to get a lot more sunlight than ones at the bottom, because the ones at the bottom are going to be shaded by the leaves above them. So when we're doing our modeling, we need to wrap our leaf level photosynthesis calculations in kind of like a broader canopy level calculation. So here what we do is we take information about the light that's incident on the canopy, and we figure out how it's absorbed and transmitted by leaves within it, so we can figure out light levels at each depth within the canopy. And so already we're seeing like a couple different disciplines being involved here. So the leaf level photosynthesis is really based on biochemistry. But now when we start to consider canopy photosynthesis, we've got some new things coming into play. Like we need to understand actually like the geometry of how a plant canopy is arranged, and we also have some like optic stuff that's that's taken from physics, where we are calculating how much light is reflected, transmitted, and absorbed by the leaves in the canopy layer. So one of the main outputs of a photosynthesis calculation is going to be the rate at which the plant is absorbing carbon from the atmosphere. But after we've figured that out, we have to decide how to relate that to the plant's growth. So some of that carbon is just going to be used for maintaining the energy levels in the plant cells. So this is respiration. So generally we need to subtract that from the carbon assimilation rate. And then the remainder is going to get distributed between different parts of the plant. So we will split it up into a couple different chunks. Here's kind of, for this example here, I'm just showing leaf, root, and pod, but other things that you might want to consider would be like the stem, or if it's a perennial plant, you might need to include the rhizome. And this is all information from plant physiology. So we're moving further away from the biochemical level, and we're moving into a plant physiology level. Another thing to consider, that we need to consider, is that the way the plant allocates carbon between its different tissues changes as the plant ages. So when a plant is young, it might devote a lot of energy to producing more leaves, so it can build up this energy generating machinery that it needs. But as it gets older, it starts switching its focus to its reproductive parts. Like for a soybean, this would be the beans. For corn, it would be the ears of corn. And this is kind of I don't know how to say this. It's kind of similar to the way a person ages. As you get older, your body shape changes. Plants are doing the same thing. For a person, it generally just depends on how old you are. Everyone hits puberty around the same age. Plants, they kind of respond to some different things. So most of them respond to temperature, and many of them also depend on the day length or the photo period. So we can use this information to predict how a plant will go through its developmental stages. As a plant gets even more old, or towards the end of its life, it begins to senesce, and it will start taking carbon from its leaves and redistributing them to other parts of its body, mainly the reproductive parts. And then the final kind of process I want to mention is that it's also very important to keep track of water. So as a plant photosynthesizes, it also kind of exhales water that originally comes from the soil. So the amount of plant growth is deeply tied to the amount of water available in the soil. At the same time, if there's not enough water available, your plant becomes stressed, and this can have a negative effect on photosynthesis. And this kind of forms a feedback loop here, which is really important to take into account. So I don't expect anyone to remember all these details, but I just want to point out that I think it's really fascinating and amazing that the work from people in a ton of different fields, from physics, from biochemistry, from plant physiology, even from atmospheric studies, the work that's been going on by all these people over decades has resulted in these different model components that we combine together in Biopro to represent the growth of a plant. And even if our models aren't perfect, I think it's really fascinating that we're able to represent them so well. And of course, an open piece of research is always, how can we improve these models further? To give you an idea, maybe one other thing I should say. So all these boxes here that I've drawn They represent model components, and they also represent equations. And what we can do with these equations is ultimately calculate the rate at which parts of the plant are growing and the rate at which soil is being added or water is being added or removed from the soil. And we can pass these equations to a mathematical solver that can determine how a plant grows throughout an entire season. And this is kind of what Biopro does for us. And I want to show an example of how that might work. And talk about a recent Biopro paper that was published regarding soybeans. So I kind of showed like a general version of a model before, on the last slide, but it can be specialized to represent one particular plant. And what you need to do that, in order to do that, is you'll need some measurements of plant mass throughout the growing season, and you'll also need the weather data. And then what you can do is you can tune some of the parameters that define the model so that they produce the right output that agrees with the experimental observations. So we call this parametrization. And here at the University of Illinois, we're fortunate to have data from a long-term soybean field experiment. It's called SoyFace, which stands for Soybean Free Air Concentration Enrichment. And this is a really cool experiment that's been going on, I think, for 20 years now, where you can actually see experimentally how soybeans will grow in an elevated CO2 atmosphere that's similar to what we expect to exist in the future. And what the experiment consists of is a bunch of these rings. So this is an overhead view of some soybeans in a field, and you can see this ring around it. The ring is actually a horizontal pipe with a bunch of little holes in it that emit CO2. And here in the very center, there's a little sensor that can measure the wind speed, the wind direction, and the local CO2 concentration. And the idea is, so if the sensor measures that the wind is blowing in from the right, then all these tubes on the right side will start emitting CO2, and it will flow over the plants. And as the wind direction and speed changes, this system dynamically updates itself to ensure that these plants here in the center are always exposed to elevated CO2. And you can see how they grow differently compared to plants that just have regular CO2 values. So something cool we can do with Biopro is try to use data just from a couple years from the ambient CO2 part of SoyFace to parameterize our model. So that was done recently in this paper, again by Megan Matthews. I'm an author on this paper too, but somewhere in the middle, so my name didn't make the cut for the citation. So the idea is you parameterize Biopro using this ambient CO2 data from SoyFace, and then you can ask, what if we increase the CO2 level in the model? Does it agree well with what we see from the elevated CO2 rings at SoyFace? And the hope is, if we go back to our model, because we use a semi-mechanistic model of leaf photosynthesis that actually takes CO2 as an input, we would hope that the soybean Biopro model responds properly to changes in CO2. And that's what we do see. So in these graphs, I'm actually showing experimental data from a different year compared to Biopro output. So we can see that in a different year where maybe the temperature and rainfall is different and the CO2 concentration is different, we still get very good agreement between the predicted and experimentally measured data. So this tells us that our model of plant growth is doing a good job of accounting for the most important environmental variables. And so what we've done is created a tool that we can use to ask other questions. So you can ask, what if we modify one of the soybean photosynthetic parameters? Now we can ask, how does this affect growth in current and future CO2 levels. And we can make these kinds of comparisons to help identify traits that could be interesting for ripe to study in actual plants. And so I think Biopro is really amazing piece of software. Part of it is these advanced models that we use for representing plant growth. The other part is the way it's written, which makes it very flexible and easy to use. And I want to say, so this is going to be the last thing I talk about, but I want to say a little bit about this. And if you'll allow me, I'd like to get a little philosophical for a second. So when I think about computer code, in one sense, it's a lot like writing things in English. Like we call English a language, and we often refer to computer code as being written in a language. And it has many like similar properties. There are things in computer code that are like nouns, verbs, adjectives, just like we have in English. At the same time, it's a bit different than regular language, because every time we write something in computer code, it's an instruction that causes an actual action or a calculation to be done. And when we write a bunch of lines of computer code, these are like little moving parts that all work together to create a big whole that accomplishes a particular task. And in that sense, a piece of computer code is kind of like a machine. And so I have two observations, one about writing, one about machines. Maybe they're not earth-shattering, but I think they're interesting to ponder for a minute. So one is that the way we write something has a big impact on how it's understood. And so as an example, you can think about whether your writing is organized or disorganized. So an organized form of writing might be broken up into chapters and sections, and kind of the extreme case of disorganized writing would be stream of consciousness, where you kind of just write down first thing that pops into your head. And there can be cases where one or the other of these is more useful to you, but generally, if you want to teach somebody about something and make the ideas as clear as possible, it's helpful to stay organized and break up your writing into chapters and sections. So the other point is that the interface to a machine can have a big impact on how it's used. And an example here where we can contrast a simple and a complicated interface would be a car transmission. So there are kind of two options out there, manual transmission and automatic transmission. And of course, there can be situations where you might want one or the other, but in general, people seem to prefer automatic transmission because it's easy to use. And there's an interesting, if you think about this a little further, like automatic transmission is not only just easier, but it actually allows some people to drive that maybe would either have an extremely difficult time driving or be unable to drive otherwise. So not only is it convenient, but it actually expands who is able to use it. And so when we think about a piece of code like Biopro, our goal is to make it organized in the way it's written and to give it a really simple interface, taking inspiration from these two examples. And I think you'll probably find this easy to believe that not all code is actually written this way. You will find lots of code that's disorganized and has a complicated interface. And so when we're working on Biopro, this is something we want to avoid, and we want to make it as easy as possible. So Biopro has actually been around for, I think, almost 30 years. But recently, we've kind of put a lot of effort into rewriting it to make it easier to use following these two principles. So this was work that was started by Justin McGrath a few years before I came here to Illinois. And then I joined him on working on it, and this has become my main project of kind of reorganizing Biopro so it's easy as possible to use. And this cartoon here is supposed to kind of explain how it currently works. instead of having to write down the equations that you use in a model, you can select them from a library of equations. So this basically takes, if you don't need to make a new model component, you can use ones that already exist that other people have written. And it cuts down the amount of work that's required to run a simulation by a huge amount. So after you choose your equations, you can supply other specifications that are needed by the model, like weather data, photosynthetic parameters, initial biomass values, and then you just send this to what we call the Biopro engine, which does all the work of combining these equations with each other and actually solving the model. And you can get the output will be the state of your plant at all the times throughout the year that you care about, with very little work done by yourself. So the key points here are, you don't need to know the details of how the equations are combined or solved in order to actually write down a model and solve it. So we think this will have a lot of positive outcomes. One is that people who might have been put off by modeling before might be able to start doing some modeling right now, because they won't need to know as much computer science or have as much math background in order to run a model. But there are a lot of other interesting benefits of this. Like for one, you can easily switch out one model component for another. Like let's say you wanted to do something crazy and just completely change the type of photosynthesis that your plant is using. This is something that would be really difficult to, well, extremely difficult to do experimentally, very difficult to do using other pieces of software depending on how they're written, but is super easy in Biopro. And I would like to give one final example of a benefit that you can get from having a modular software for crop growth modeling. So this, for this example, I'm going to talk about how this kind of flexibility can help you integrate data from sensors with a crop growth model that already exists. So to make this more concrete, in a typical Biopro simulation, we use like a leaf area equation, which calculates the area of the leaves from the mass of the leaves. We just use this simple equation where we multiply the mass by the specific leaf area to get the area of the leaves. And a lot of times this will just be considered to be a constant value. And you're saying like the leaves have a certain density throughout the entire year. But what if instead of using this model, you suddenly had access to leaf area measurements? Now you wouldn't need to make this assumption anymore that area and mass are related by a constant value, and instead you would like to use your actual measured values of leaf area. So depending on how your model code is written, this could be extremely difficult to actually do. But in Biopro it's quite easy, because all you do is just stop using this leaf area equation when you're calling Biopro, and instead you can supply your measured values of leaf area. So in this case, the result of doing this, it might let you see how the ratio of area and mass for the leaves of a plant change throughout a year, instead of assuming a constant value. And it could be used for developing a new, more accurate model. And this ability can also be really critical for kind of a growing field that's called smart agriculture, where either researchers or farmers are including many sensors out in their field and monitoring as many things as they can. So you could imagine using that data in real time as an input to a Biopro model and making predictions about what your plants are going to do during the rest of the year. And I want to point out that we're expecting some like interesting developments in this area here at UIUC, and they're related to a new tool that we have available to us experimentally, which is really awesome. It's called the Spider Cam. So this is the same technology that's used at sporting events to move cameras around the field. Like if you watch a football game and there's a camera overhead, like following a player or the ball or something, it's probably on a spider cam like this. And the way it works is there are big towers, there are 4 of them, with wires coming off from each one that are attached to a platform. And by pulling and loosening on the wires variously, you can control very accurately where it is over a very large area. And you can strap whatever kind of measuring instrument you want on here. So something that we're going to work on in the future is adding a 3D scanner here. So a LiDAR scanner that can give us a 3D model of plants. So you could imagine passing this thing over your plants that are growing in the field, and you can actually directly measure their leaf area. There are other things that you can measure, and it's not actually that simple either, because you don't measure leaf area. You measure like a complicated 3D model of a plant, and then you have to figure out how to extract leaf area from that. So there are a lot of challenges, but it's definitely like an exciting new development where we think that improvements in our software are going to synergize very well with improvements in our experimental measuring techniques. And to kind of summarize what I wanted to say about Biopro, we kind of have like a vision of the future of computer modeling, where which is kind of summarized by this cartoon. So on the left, you have a model consists of different parts, and it represents the status quo for some particular crop. It's predicting a yield of this many beans. And then along comes a researcher who has an idea for something that they think they would like to change in the way that this plant works, and they're hoping that it produces a larger yield. So they're able to go through using Biopro and just make a small change to one component of the model, or maybe add a new component, and see how it affects the yield. And here, this researcher got lucky, and the model is now predicting a big yield, so this could be something that's worth trying to move into a real plant. And we really hope that it will be almost this easy in real life to do. Yeah, so to conclude, today I wanted to tell you about how plant biology is playing a key role in ensuring food and energy security in our changing climate. I wanted to tell you how RIPE is working to increase crop yields and is committing to sharing technology across the world. I wanted you to learn that modeling is a critical part of this whole process, and it's required to guide and interpret other experiments. And finally, that we are working on Biopro, which is a flexible crop growth modeling tool, and we hope it will enable lots of people to do new types of computational studies much easier than they used to be able to. And there are a bunch of people I would like to thank. My advisor, Steve Long, my co-worker / sort of advisor, Justin, and various other people around the department who have helped me with my work or just in understanding plant biology better. And with that, I will conclude my talk.
Awesome. Thank you very much, Ed. It was amazing talk. If anyone has any questions, feel free to either unmute yourselves, or you can throw them in the chat if you don't want to unmute and talk yourselves. But and yeah, I did have a couple questions. My naiveness on modeling. So you mentioned that like Biopro is an R package. Do you know what is an R package? Can you?
Okay. Yeah. So R is like a free piece of software that lets you do lots of like. It's hard to describe what it is because it's so like open-ended, but it lets you like manipulate data or like perform analyses of data to create plots, to perform statistical analysis methods, and by including Biopro within R, we basically give you all of R's powers for like reading it and accessing data and plotting it and performing analyses. So it kind of it combines with Biopro's built-in abilities, and it also makes our work easier because if we put Biopro in R, now we don't have to worry about providing methods for people to visualize their data. They already exist.
Awesome. Very cool.
It's kind of like it's like MATLAB or similar tools like that, if you're familiar with any of those.
No.
Okay, I see this in the chat. Any thoughts on how to incorporate genetic information into Biopro models? Yeah, this is a great question. It's very complicated, I would say. Right now, there's not a way to, not a good way at least, to directly incorporate genetic information. You can do something like, say, I predict that this particular genetic change might cause a change in the photosynthetic parameter like VC max by increasing it 20%, and that would be something that you could include in a Biopro model. In general, the path for including genetic information would just be to you would need to create a model component that relates it to something like more observable. Maybe along the lines of what I mentioned with the lignin poplar study. So a lot of work went into creating a model that can relate genetic information to like a larger scale property of a plant. But if you had something like that, that would be relatively straightforward to put into Biopro. Does that answer your question? Basically, it's not impossible, but it's very complicated. It's definitely something that people are interested in, incorporating genetic information into crop growth models like this. I'm not really aware of it having been done before.
Another question that, Justin has something here. Oh, cool.
I get another thing just occurred to me that I remember. Generally, it's possible to represent like a gene network mathematically using a system of differential equations that explain the rates at which like mRNA is transcribed from DNA, and then the rate at which proteins are created from the mRNA. So if you wanted to, you could include that as a component of a Biopro model. And then I think the time scale at which you would be solving it might be a lot different. Like right now we typically take like 1 hour steps, but that might be too large of a scale if you want to include these small scale genetic gene network properties. But actually something like that approach has been attempted before for Arabidopsis, not using Biopro, but many of its at least some of its properties, like its circadian clock, were represented by a differential equation system that represented a gene regulatory network. I could send a link to that if I can find it.
And yeah, and it looks like Justin threw in the chat also a link to a study where they did something similar, incorporating gene networks into leaf level models. So it's very interesting. Another question that I had was more of a general thing, like when you're doing these simulations and these models, have you gotten any like surprises, like things that you modeled that you would have expected to have a significant impact onto plant growth or photosynthesis that didn't have as big an impact as you expected, or the reverse, like something minor you changed that had a bigger impact than you were originally thinking?
Well, maybe before I attempt to answer that, I should tell you, maybe give you a little bit more info about what I typically do. Um, I haven't actually used Biopro for scientific purposes very much because I've been very focused on the development side. And with the publication of the paper that I cited towards the end, we're kind of getting to the point where we think that the main parts of Biopro development are done. And I'm hoping to start using it for more scientific purposes. Another thing I should mention is that, well, if you want to ask me, like, have I personally found anything surprising, the answer is going to be yes, and it's related to the fact that I've only been a plant biologist for a couple of years. So lots of things might be surprising to me that maybe not are surprising to other people. But one thing that I recently learned about and was able to demonstrate for myself in Biopro, so I actually used the models that are in Biopro to teach myself about plant biology a lot. But anyway, something I learned about was the fact that it's actually possible for a crop to produce too many leaves, in the sense that as it produces more and more leaves, the ones at the bottom start receiving less and less sunlight, but at the same time there's still a cost that the plant has to pay for maintaining those leaves, because all the cells in those leaves are consuming energy, and if they're in very dim light, they're not really producing any. So it's actually an interesting thing that some plants make too many leaves, and soybean is one of them. And so you can actually see this in the soybean Biopro model. One cool thing we can do with Biopro is isolate just a piece of it. So I can take out like just the Canopy Photosynthesis model, and I can say, what's your carbon assimilation rate if there are so many leaves? And I can do that for many different values, and I can see that there's kind of a peak. There's like a perfect amount of leaves, and when you start adding two more or too many, the photosynthesis rate goes down. And that was pretty interesting to me. And you can kind of like keep going with that kind of line of thinking, and once you see something like weird like that, well, at least it was weird to me, like you can say, you can look around in other parts of the model, and so you could look at like the partitioning part where carbon is allocated to the different organs, and you can say, like, what if I told the soybean to put less carbon into its leaf production? How would that affect its final yield? Like, maybe because this plant is making too many leaves, if I tell it to make less, it could have a positive impact on yield. And that's something else that you can see that in the results from Biopro that I thought was pretty cool. I don't know if Justin might have anything to say about an unexpected result from Biopro, because he has actually used it more than me.
I mean, I don't, yeah, let's see my kind of messy house now. One of the things that I often find surprising, it's not just from Biopro, but from real plant biology studies and just modeling in general, is if you look at the advances we've made in crops and we know what has caused that improvement. Like, so we yield is much higher yielding, or sorry, maize is much higher yielding than it used to be. Soybean is much higher yielding. And when we try to understand why that happens, for the cases where we know, it's always decreased competition between plants. And so maize used to have very flat leaves that kind of came out from the side. And then that would mean they absorb a ton of light, but they can't use it actually. Once they get so much light, it just gets wasted. That was that NPQ that Ed was talking about earlier. And so they do that because they can shade their neighbors. So even if they can't use that light, it's good for them to keep their neighbors from getting it. So modern maize cultivars, their leaves go almost straight up. And so this lets them, that plant will absorb less light, but now it's no longer absorbing more light than it needs. Then you can plant them closer together. So we get a higher maize yield, not so much because each corn plant has more yield. It's because you can plant them closer together. And so if you look at maize roots, they used to be very shallow. So they're trying to steal their neighbor's water. Now they're very deep. And so you can see these same sort of things in modeling. Like, so a recent modeling study found that leaves probably have too much chlorophyll. So it's the same idea. They have a ton of chlorophyll to absorb light that keeps their neighbors from getting the light, but then they can't use it. And so modeling has identified that as well. And people are actually trying now to create low chlorophyll crops. And so when you look at them, they don't look good. To us, they don't look good. We're used to seeing green plants. These look yellow, but they don't seem to have a yield cost. And so it seems like it's a benefit. There'd have to be you have to do something else too. Like with maize, making the leaves straight, that doesn't improve yield. Making the leaves straight and closer together, that's what improves yield. So this is going to be the same thing where you can decrease chlorophyll and you'll have to do something else too to improve yield. And that came from modeling. So, people had not thought of that before. And that's where it's something to identify that's completely unintuitive.
Yeah, that's really interesting to see. I would never have thought plants have too much chlorophyll or anything like that. That's really cool. Yeah. Well, if there's no other questions, Ed, thank you so much for the super interesting talk. I'm really excited to be able to listen and see everything else.
Yeah, thanks for inviting me. I had a fun time giving the talk.
Yeah. Awesome. Well, I hope you all can join us next month for the last Science Cafe of the semester. But otherwise, have a great day, and thanks you all for joining us.